Heart-on-a-chip breakthrough promises faster, safer drug testing for heart disease. Scientists have engineered a three-dimensional heart-on-a-chip (HOC) that mimics human heart tissue with unprecedented accuracy, potentially revolutionizing how cardiovascular drugs are tested and how heart disease is treated. Cardiovascular disease is the #1 killer worldwide, and this technology addresses a critical bottleneck: the difficulty of safely testing drugs and understanding heart responses without risking human lives. The problem with current testing methods traditionally, assessing how a drug or disease affects the heart, requires either animal models, which don't always translate well to humans, or clinical trials with real patients. This new HOC offers a middle ground: a functional, beating heart tissue engineered in a lab that can respond predictably to medications. The key innovation is its ability to monitor activity at both the tissue-wide and cellular levels in real time. Previous HOCs lacked this high-resolution sensing, meaning they couldn't reliably detect subtle changes in individual heart cells. A crucial factor, since many heart diseases begin with dysfunction at the microscopic level. How the heart-on-a-chip works: the research team from Canadian institutions created the HOC using cardiac muscle and connective tissue cells harvested from rats. These cells were embedded in a growth-promoting gel matrix and seeded onto flexible silicon chips. The system incorporates two sensor types: macro-scale sensors. Elastic pillars deform with each heartbeat, measuring overall contractile strength. Micro-scale sensors, tiny hydrogel droplets, 50 micrometers in size, capture local mechanical stresses at the cellular level. This dual sensing platform allows scientists to see how cell-generated forces influence tissue behavior, including growth, healing, and even cancer progression. The team successfully demonstrated the HOC's functionality by treating it with norepinephrine to increase heart activity and blebbistatin to decrease it, both of which produced the expected responses. Why this matters? Precision medicine for heart disease, this breakthrough has immediate implications for drug development. The HOC can now be used to screen compounds before human trials, accelerating the process and reducing risk. More importantly, it opens the door to personalized medicine, the ability to test a patient's own cells against various treatments to identify the most effective option before prescribing medication. This breakthrough brings us even closer to true precision health, says Human Saivoji, a lead researcher. By giving us the ability to identify the most effective medication for each person before treatment is even administered. Future directions: the team plans to simulate specific cardiac disorders, such as dilated cardiomyopathy and arrhythmias, by using cells from patients with those conditions. This will allow for more realistic testing and potentially lead to targeted therapies. The heart-on-a-chip represents a significant step toward a future where heart treatments are tailored to individual needs, improving outcomes and reducing the global burden of cardiovascular disease.